Hello everyone. Today, it's my pleasure to introduce MicrobTiSDA, a flexible R package for inferring interspecies interactions and abundance dynamics in microbiome time series data. Microbiomes are ubiquitous, existing not only in the human body but also in diverse environmental habitats, and they are highly dynamic. The species composition within microbiomes continuously changes over time, influenced by numerous external factors such as diet, disease, antibiotics, and other environmental perturbations. Consequently, microbial time series data analyses often aim to characterize the temporal abundance patterns of microbial species. Moreover, a microbiome can be viewed as a microecosystem, in which species interact with one another and collectively shape the overall community structure. These interactions may be either positive or negative. Therefore, in the analysis of microbiome time series data, it is equally important to elucidate the interspecies interaction networks that govern community dynamics. MicrobTiSDA is a functionally modular R package comprising 7 core modules. It begins by accepting microbiome time series data, associated metadata, and taxonomic information as input, which are then processed through the preprocessing module. Following preprocessing, the species interaction inference module, on the Lotka-Volterra model, can be used to infer interspecies interactions within the microbiome. In parallel, the regression modeling module allows for fitting the temporal abundance patterns of individual microbial features and clustering features with similar temporal dynamics. Additionally, for group-level studies, MicrobTiSDA provides a random forest classification module to identify key microbial features that effectively discriminate samples across different experimental groups. The 2 core functionalities of MicrobTiSDA: constructing regression models to predict the temporal dynamics of microbial feature abundances and inferring interspecies interactions within microbial communities, were systematically evaluated. By comparing MicrobTiSDA with 3 other similar tools in terms of regression model fitting performance, we demonstrated that the natural spline regression approach, implemented in MicrobTiSDA, provides superior performance in modeling the temporal dynamics of microbial abundances. To evaluate the performance of the species interaction inference module, we tested MicrobTiSDA using simulated datasets with known interspecies interaction coefficients. The results indicate that MicrobTiSDA can accurately infer the interspecies interaction coefficients in the simulated data. In addition, we assessed the computational efficiency of MicrobTiSDA, and the test showed that the tool achieves rapid runtimes when the number of species in the dataset is relatively low, such as fewer than 500 species. MicrobTiSDA has been applied to 3 microbiome time series datasets with distinct experimental designs and research objectives: an in vitro aquatic microbiome dataset, a longitudinal dataset tracking gut microbiota changes in full-term infants during their first year of life, and a dataset capturing gut microbiota dynamics in preterm infants the first month after birth. These applications highlight the flexibility and broad applicability of MicrobTiSDA in handling diverse microbiome time series data. In summary, we present MicrobTiSDA, an R package specifically developed for the analysis of microbiome time series data. The package provides a streamlined and fully integrated workflow covering all major analytical steps, from data preprocessing to result visualization. Compared with existing tools, MicrobTiSDA demonstrates superior performance and computational efficiency, as well as remarkable flexibility and broad applicability. The installation package and user guide of MicrobTiSDA are publicly available on our GitHub repository, and we warmly invite researchers to explore and utilize this R package. Thank you.